Okay, are there any questions? If there's no questions, let's go ahead and get started.
I do have a question.
Okay, go ahead and ask.
For the unknown project, you know how we have that key for the lab this week? Do we have access to something similar like that for our unknown project?
Can you repeat that? I didn't quite understand.
For the unknown project, how we have a key this week for the lab, lab 8. Is there something similar we can use to identify the unknown bacteria?
There's no key, but there's a table. The table of the unknown list.
Okay.
You're supposed to make a key for your unknown project, or not the key, but the path you use for identifying your unknown. So you will make one in your unknown report.
Okay.
Okay.
Thank you.
All right. Let me plug in my headphone. Good. I didn't take out of there, so I should be able to remove this. Didn't quite have my headphone ready. Any other questions? Now, let me remind you that you don't want to wait on your unknown project to the very end because it'll take time. Supposed to be putting all your requests in the unknown notes page. All right, and I guess I'll emphasize that the project should be following dichotomous logic. All right. Today is May 12th, and we should be starting chapter 8 today. We're a little behind schedule. I hope to get started on chapter 8 today, but we need to finish up chapter 6. Today in the lab, we have lab 8. Any questions about what we're doing? All right, so let's get started. Make sure I'm recording. Yep, it is. So we had talked about the 4 toxic forms of oxygen last time. Singlet oxygen, superoxide free radicals, peroxide anion, and then the hydroxyl radical. Any questions about the 4 toxic forms of oxygen? You always have these 4 toxic forms of oxygen whenever oxygen is around. And it's true that oxygen itself is not toxic, but some metabolites of oxygen are. And the anaerobes are actually killed by these toxic forms of oxygen because they don't have the enzymes to protect themselves or the antioxidants to protect themselves. All aerobes have the enzymes and the antioxidants to protect themselves from oxygen. So oxygen is a requirement for growth, but some organisms require that there be no oxygen. And I don't know how you want to word that, but the oxygen is a requirement, and Some organisms require that oxygen be absent or they will not grow. In fact, they will usually die if oxygen is present. Microbes that use oxygen, we call them aerobes. They produce more ATP from nutrients than those microbes that do not use oxygen, and we call those anaerobes. Microbes are classified on the basis of the effect oxygen has on their growth, and you can look at the growth of microorganisms by growing it in a thioglycolate tube. Thioglycolate, when you make the media and you autoclave it, the media pulls oxygen out of the media. So it has something in the media that reacts with oxygen and then removes the oxygen. Any question about any of that? But that's how the media is when it first comes out of the autoclave. The trouble is, oxygen is in the air, and assuming this cap is loose, oxygen can come into the tube, and then we will have an oxygen gradient in the thioglycolate tube. Where the heck is thioglycolate on here? I don't know. So at the top of a thioglycolate tube, you'll have full oxygen, 20, 21%. And as you go down the tube, you have a gradient of oxygen, and the oxygen will decrease until you get to the bottom of the tube, where oxygen will be either nothing or nil. If all of the growth of the microorganisms only grow at the top, and as you go down the tube, you see less and less microbes, then this microbe is an obligate aerobe. Any question about any of that? If, on the other hand, you look at a thioglycolate tube and the microbes are only growing at the bottom, and as you grow up the tube, there's fewer and fewer microbes. And what we should do is have one dot right here to say that there's a little bit growing in this region, but none above it. Then you'd say this is an obligate anaerobe. It can only grow where there is no oxygen. And as you go up in the tube, there will be less and less growth until such a time where there is no growth. There are also organisms which we call facultative anaerobes, and they can grow in the presence of oxygen, and they can grow in the absence of oxygen. So you'll see the growth everywhere in the tube. However, if oxygen is present, they will perform aerobic respiration. And as you know, aerobic respiration is more efficient than anaerobic respiration. So they will have the most growth at the top of the tube because they can make more ATP with aerobic respiration than with anaerobic respiration. And so you'll see the growth a lot at the top, a little less below that, a little less below that. And at the very bottom, we should have only one dot so that there will be less and less growth the further down you go in the tube. But the growth will occur everywhere in the tube because they can perform aerobic respiration if oxygen is present. But when oxygen isn't present, they will perform either anaerobic respiration or fermentation. Any question about any of that? There are also aerotolerant anaerobes. They are anaerobes, so they do not need oxygen to grow. But the aerotolerant anaerobes have all of the enzymes and antioxidants that they need to protect themselves from oxygen. So they can grow uniformly in the thioglycolate tube, meaning they can grow in the presence of oxygen, and they can grow just as well in the absence of oxygen because they are an anaerobe. Any question about any of that? And then there are also microaerophiles. These are aerobes. They do require oxygen to grow. They will not grow in the absence of oxygen. However, they do not grow well, if at all, in full oxygen. They grow better at reduced oxygen concentrations. So the majority of the growth will happen someplace in the middle of the tube. The microorganisms in your stomach are actually microaerophiles, like Helicobacter pylori. They are aerobes, but they grow best at reduced oxygen concentrations, usually somewhere around 2 to 4% oxygen is where they have maximum growth. Any question about any of that? All right. Now, obviously, if in your unknown project you were to do the thioglycolate tube, which I don't think we have that in the table, but there would be more than 2 possibilities, but you could still do one test at a time and follow dichotomous logic. All right, if there's no questions about the effect oxygen has on growth, let's talking about culture media again. We've already talked about general purpose media, and let me state that media is plural. If you're talking about one, then you use the word medium. Plural is media. Mediums, or I guess it's media, are either liquid, we call those broths, or they're solid. They have an additive added to the broth, it's usually agar, to make it solid. Media contain all of the nutrients needed for microbial growth. The media given to you in the lab, if you were actually go to a lab, is sterile. And how it becomes sterile is it was put in the autoclave. That means there are no living microbes present if it's sterile. An inoculum is when microbes are introduced into the media. The culture is what we call microbes growing in the medium. So if you spill the culture, that means you're spilling the media that contains microbes. There are a very wide variety of media available for microbiology. If you were in the lab, I'd show you a book. It's about this wide and that long, and it's about that thick. On each page of that book, there's about one media. Okay? So that tells you how many different media we have in microbiology. Any question about any of that? Now, we won't obviously talk about all those different types of media, but media need to support microbial growth. A media must provide an energy source, and that's usually something like protein or carbohydrates, like glucose. And it must provide a carbon source. That carbon source is often glucose or a protein. There must be a source of nitrogen, sulfur, and phosphorus. And if you're including protein in the media, that could be the source of nitrogen, sulfur, and not really phosphorus, but certainly nitrogen and sulfur. The media must contain trace elements, and oftentimes the trace elements just come from the water you use to make the media. But the trace elements can also come from the beef extract, meaning they dry the beef, grind it up, and make a powder out of it, and that can supply the trace elements. If the media requires organic growth factors, then that has to be added to the media. I don't think I said that right. If the bacteria you're growing requires organic growth factors, then the organic growth factors have to be supplied by the media. Now, we've already mentioned that Streptococcus pyogenes and Haemophilus influenzae require organic growth factors. And those are supplied by the red blood cell that's added to a blood agar or chocolate agar. So if the organism needs these sources, it has to be supplied by the media. But what does a photoautotroph that fixes nitrogen from the air, what would this organism require in the media?
Nitrogen?
No, it gets nitrogen from the air, so that doesn't need to come from the media. Does it require an energy source? It's a photoautotroph that fixes nitrogen. What does it require? Does it energy source?
Light?
Yeah, it doesn't need the energy source from the media because it can get its energy source from light. And it's a photoautotroph, so we don't need to supply a carbon source in the media because it can pull carbon from CO2, meaning it's an autotroph. We would need to supply sulfur and phosphorus and trace elements in the media. Photoautotrophs generally do not need organic growth factors because they can make their own organic growth factors. But if we were growing your cells in a media in the lab, which we would never do, but if we did, we would have to supply an organic growth factor. And can anyone remember an organic growth factor you have to supply in a media to grow your cells?
Is it blood?
Say again?
Is it blood?
That would supply the nutrients you need, but let's say you're not putting blood in because you're growing your own cells. And if you were to put blood in, that would contaminate your cells with the blood cells.
Water?
What's something you would need to add to the media to grow your cells? Okay, you would have to.
Would it be glycogen?
Like, say again?
Would it be like glucose?
No, that would be a carbon source. You have to supply something like all the vitamins. I was hoping someone would remember vitamin C is an organic growth factor, at least for human cells. Now, if you were growing mice cells, they can make their own vitamin C, so you wouldn't need to supply that. But human cells need vitamin C. So generally all of the vitamins and the essential amino acids would have to be supplied. All right, agar is something that's added to media to make it a solid agent, to make it the solid media. An agar is a complex polysaccharide derived from seaweed. Agar is usually used in Petri plates or in tubes. Let me see if I can bring up paint here to show you a tube. Tubes are usually slants where the media is something like that, but they could be where the media is something like that. That's not very good. Let's try that again. That's even worse. I'm trying to draw a straight line across. No, that's about as good as I'm going to get. Sorry, I'm not an artist and that. So we call this a deep and that a slant. And you could have them combined where you have the deep and on top of that you have the slant. Oops, that's not very good. And then you don't see that there, so that'd be like that. So they could be combined like that too. But those are the two ways that auger are used in a tube, a slant and mostly a deep. And that's close enough. I should have made that a steeper angle. Auger is generally not metabolized by microbes. Why it is used so much in microbiology to make the media solid. If you read about Dr. Koch, he invented growing things on solid media and initially made the Petri dishes solid by adding Jello. But Jello can be digested by many microbes. More of them do not, but there's a large number of microbes that digest the Jello, and then the solid media would become liquefied. And so that's why he switched to auger. And auger was actually suggested by the wife of his lab assistant, which I think was Hess. I think it was Mrs. Hess, who suggested, why don't you use auger if Jello isn't working? An auger, if you don't know, is something that you can use in baking or in cooking that solidifies, well, food. There are a wide variety of different culture media. One we're going to talk about is chemically defined media. This is a media where the exact chemical composition is known because all of the additives to the media are added by the person who makes the media. A very simple chemical media is one containing glucose that supplies carbon and energy, ammonium phosphate supplying both nitrogen and phosphate, sodium chloride, magnesium sulfate. The magnesium sulfate supplies the sulfur, and then potassium phosphate, which supplies more phosphate. And then you just add water. So all of the ingredients of this media we know because it's distilled water, meaning there's no trace elements added. All of the ingredients in this media are known, and that's the chemically defined media. This is often used with autotrophs because they're not very picky, and you can grow an autotroph in a media like this. I used to make a glucose media, glucose salt media, which would be similar to this. Now, a fastidious organism could not grow in a media like this because it needs a lot of requirements added. And this would be a media that is chemically defined, but for a fastidious organism. So we're adding a large number of different carbon and energy sources, a large number of salts to supply phosphate, sulfate, magnesium, nitrogen, calcium, iron. And then we're supplying some amino acids. These would be growth factors. And then organic growth factors right here. Thiamine, that's a B vitamin. Nicotinamide, another B vitamin. Biotin, another B vitamin. Uracil is a nucleotide. And then this is adding sodium thioglycolate. So this would be growing an anaerobe. Okay, any question about that? The point is that this is still a chemically defined media. We're using distilled water. So we know all of the additives that are in this media. There are also complex media, and these media contain extracts that are usually digests or extracts of yeast, meat, or plants. For example, this media here, which is nutrient agar, contains peptone, which is a partially digested protein, beef extract, so they just ground up the cow and made a powder of it. And then they add sodium chloride to make the ion correct, and agar to make it solid media, and then just water. And if they're using distilled water, that's it. However, the beef extract and probably the peptone will be adding trace elements. And we don't have any idea what all the molecules are in this media, because we have no idea what the cow was eating. And so the cow could be adding a whole bunch of different molecules. Okay, any question about complex media where the exact chemical composition is not known, but we are providing a carbon source? That would be the peptone and the beef extract, a nitrogen source, also the peptone and the beef extract, and a sulfur extract. That would be both the peptone and the beef extract, because both will have protein in it. We use complex media usually to grow heterotrophs, organisms that require an organic carbon source. And like for E. coli, we can grow that on nutrient agar. Now there is a different type of a media, and I've already mentioned it, and that is anaerobic media. This is a media for growing obligate anaerobes, organisms that must avoid exposure to oxygen. An anaerobic media has a reducing agent in the media, meaning it has a chemical like sodium thioglycolate that combines with and removes oxygen. And that way, oxygen will be depleted, at least at the bottom of the sodium thioglycolate tube. What you do is you heat the media before you use it, and when you heat the media, it drives off any absorbed oxygen that was in the water or in the media when you made the media. And then the sodium thioglycolate will pull out the oxygen from the media, at least at the bottom of the tube. Any question about any of that? There are different types of media. There are selective media. This is a media that has a chemical in it that halts or inhibits the growth of a particular group of microorganisms, but does not affect the growth of another type of organism. For example, if we put 7% sodium chloride in the media, only organisms that could tolerate and grow in 7% sodium chloride would be growing in that media. So it's selective for media or organisms that can tolerate 7% sodium chloride. If they do not grow in 7% sodium chloride, they will be selected against. Any question about selected media? There are also differential media. They have chemicals in it which provide differences between two different types of organisms. The differences are not related to how well the organism can grow on the media. Usually, the chemical makes the two types of cells or two species look different by either color or, in this case, by what's happening around the growth of the cells. Blood agar is an example of a differential media. You can see three different results on blood agar. The cells could perform gamma hemolysis, and that's no hemolysis, where the blood are intact in the media all the way up to where we have the cells growing, the white Greek letter gamma here. Any question about any of that? The blood agar can also show alpha hemolysis, and that is shown here where the white alpha is where the cells are growing, and the dark ring around the growth is where there is partial hemolysis, and we call that alpha hemolysis. The cells are partially lysed, and it makes the blood look dark or greenish. And that's that dark ring around the alpha. It's also the darkening inside the alpha. The third possible hemolytic reaction is beta hemolysis or complete hemolysis. And that is where the red blood cells are totally lysed. We can see the white beta is where the growth is. And then this clear area, that's not white, that's clear, where the light is actually shining through it. That's where all of the red blood cells have been lysed around the beta. Okay, you can actually look through or see through the media. So if you were to look through this right here, you'd be able to see through wherever it's clear. Any question about the three types of hemolysis? Okay, you normally see a ring of alpha hemolysis around beta hemolysis. So that is normal. Any question about hemolysis or blood agar? The point is that's differential because we can see three different types of cells where they're having different hemolytic activity. There are also selective and differential media. Eosine methylene blue or EMB plates are selective and differential. They are selective in that they select against gram-positive bacteria. Generally speaking, if something is growing on EMB, that means it's a gram-negative cell. It's also differential on the basis of lactose fermentation. If it looks black, the growth, or greenish, and that looks like metallic greenish, then it is fermenting the sugar lactose. And depending on the how much fermentation goes on, it will look a different color. Okay, the greenish or blackish is fermenting the sugar lactose. And if it's not greenish or blackish, generally the same color as the organism, it does not ferment the sugar lactose. So this organism, which looks more or less white, does not ferment the sugar lactose. And if it's dark, or black or greenish than it does ferment the sugar lactose. So it is both selective, growing only gram negatives, and differential on the basis of lactose fermentation. There's also an enrichment media. An enrichment media encourages the growth of certain organisms. So therefore, it is a selective medium, but it is used to enrich the organisms of interest. And so I guess you could call it a special category of a selective media. It increases the numbers of the desired microbe to detectable levels. It's particularly useful if the targeted microorganism is only a small proportion of the total population. And it's often used when people are trying to isolate specific organisms growing in the soil or other environmental samples, or, for example, a fecal sample. Suppose you have a clinical specimen and it's a fecal sample and you're looking for a human pathogen where there are large numbers of microbes normally present in the feces. And the pathogen may be at very low numbers. And so what you do is you grow it in the enrichment media, and then you can actually have the chance of isolating your human pathogen. Let me talk about that in a little different example, and that is we're looking for an organism that digests phenol, and we're looking at a soil sample to try and get the phenol degrading bacteria. Let's say that there are thousands of other bacteria. If you were to try and streak out the organisms that you isolate from the soil, then your chance of finding it would be 1 in 1,000 or maybe 1 in 10,000 colonies. So that your chance of finding the phenol degrading bacteria would not be very good. What you could do is take your soil sample and grow it in an enrichment media. If it's enriching for phenol degrading bacteria, such as a media where phenol is the only carbon source, as well as the only energy source, that would enrich for phenol degrading bacteria. So let me start here again. Oops, wrong one. So we're going to pretend this is now a liquid media. We put the sample in the tube here, and let's say it only increases 10 times. So that the initial chance of finding it was 1 in 10,000 cells. Oops, didn't mean to do that, that's fine. So we grow it in the enrichment media. Now our chance of finding it will be 1 in 1,000. That's still not very good. You'd have to sample 1,000 different colonies to find your organism. But let's take this and put it in another enrichment media. If it increases the concentration tenfold, we've now got it to 1 in 100. This would be difficult to find still. You'd have to isolate 100 colonies before you get your colony of interest, but it is doable. Still, what you'd want to do is transfer it one more time into another enrichment media, and we're just assuming it's increasing at tenfold, always. And now we're going to have the cells that we're interested in, 1 in 10. All you have to do is sample 10 colonies and you would get 1 colony of interest. You could do this in 1 petri dish, 1 petri plate, sorry. You could easily get 10 colonies on 1 petri plate and then just isolate 10 of them and sample, and 1 of them on average would be the organism of interest. And that's why we use enrichment media, to enrich for an organism of interest. Any question about any of that? All right, so this slide shows you the difference between a chemically defined media, a complex media, a reducing media, a selective media, a differential media, and an enrichment media. And please note that the media can be more than one. We mentioned there's selective and differential media, and then I showed you blood agar, right? Well, this is both a differential media, but it is a enriched general purpose media, meaning an enriched media. Now, note the difference between enrichment media, that's a selective media, and an enriched media. An enriched media we just use to grow fastidious organisms, organisms which are difficult to grow. Okay, so there is a difference between enriched and enrichment. Any questions about that? All right. A pure culture is a culture that contains only 1 species. How you get a pure culture is you streak your cells through 2 colony isolation events. And then you will make the chance that any colony is at least 95% pure. So what you do is you streak it out for colony isolation 2 times. The first time only increases it to 70%, but if you do it again, you'll increase it to 95% or higher, that a colony will be a pure culture. A colony is a population of cells arising from a single cell or from a clump of cells, like 2 or more cells stuck together. Therefore, when we're talking about colonies on a plate, we call the numbers of units that form the colony forming units, because a colony may or may not be formed by 1 single cell. It could be formed by 2 cells or from 3 cells or even a clump of cells. So to obtain single colonies, it's a critical part for obtaining a pure culture. And what you do is you simply streak plate your cells twice, streaking for colony isolation twice, to make sure that you have a pure culture. And even then, you should remember that it may not be pure. It's only 95% or higher that it is pure. Any question about any of that? All right, this is showing you how to streak for colony isolation. Generally, what you do is you do in sectors, and you streak 1 sector from your stock culture, and then you flame the loop, and then you streak the other sector by picking up cells, like we're starting over here. And then we pick up cells from the previous sector, and then carry those cells into the new sector. And then we don't go and pick up more cells. So we only pick up cells a few times, usually 3 or 4 streaks is what you do to pick up the cells. And then you come and then you streak this sector. And then you streak this sector. And once again, you sterilize the loop. And then you start in sector 3. And then you'd move this way, picking up the cells. And then you streak over here. And you don't go back into that sector. Okay. Streaking for colony isolation is very good if the organism you're looking for is high in frequency in the population. If it's really rare in the population, you should grow it in an enrichment media before you streak out for colony isolation. And as you can see here, when we streaked out the cells, we saw that we had a mixed culture. There are mostly red colonies, but there are a few yellow colonies. Okay. And what we do to make sure we have a pure culture is isolate from a single red isolated colony and then streak it out again. And then to set up our stock culture on the second plate, we would take the cells from one colony, meaning it's gone through 2 colony isolation events, and then streak that out on a plate to set up our stock culture. Any question about any of that? All right. The growth of bacteria usually happens by binary fission, where one cell splits into 2. But there are a few species that actually grow by budding, where a cell forms as an outgrowth, a bud of the parent cell, and then that bud grows, it enlarges. The bud will eventually separate from the parent when the bud grows to almost the same size as the parent cell. There are also a few bacteria that grow by reproductive spores. Let me see if I can show you that again. Where we have our parent cell, and usually it will be a large cell and it'll go up like that. And what it does is it forms reproductive buds, usually in this region. And then it's waiting for the wind or something to pick up one of these buds and then carry it elsewhere so that the bud, not the bud, the spore can grow in a new location and then form a colony over here. So that's reproduction because one cell made, in this case, 4 spores. And we're going to call these reproductive spores to separate them from endospores. Remember, an endospore is not a reproductive structure. One cell forms one endospore, and then the parent cell dies. Any question about any of that? So here we're looking at cell division by binary fission. The DNA first divide, and then one molecule moves to this side of the cell, the other molecule moves to that side of the cell, and then this cell literally splits in 2. It forms both a cell wall and a cell membrane in between the 2 nuclei, and the cell divides in 2. Right here, we can actually see a cell wall growing in between the cell in an electron microscopic image. And then that cell will divide into two. And then these cells can be isolated and go their own separate way. Any question about any of that? I've learned that my link from PowerPoint won't open, so I'm just copying it. Let's watch it. Cell budding, where one big parent cell buds into a young baby cell. So here we see a bud starting to form right there. And there the bud is growing. And there you can actually see the DNA moving into the bud. And there's DNA there and DNA there. All right, so now this cell is about the same size as the parent cell. It's now separated from the parent, and this parent's gonna make another bud there, and this cell is gonna make a bud here. Any questions about budding? Nope, I didn't want to do that, but that's all right. All right, when we're talking about growth, it's important to discuss the generation time. Students often have a problem with the generation time. The generation time is the time required for one cell to divide and become two, and that is the same thing for the population of cells to double in their size. The generation time varies greatly between species. It can be as fast as 20 minutes, but most bacteria take from 1 to 3 hours to double, meaning their generation time is 1 to 3 hours. But Microbacterium leprae is extremely slow. It takes 10 days for one cell to divide into two, or for the population of M. leprae to grow or to double in size. Mycobacterium leprae causes leprosy, and it's very slow, partly because of all the macolic acid in its cell wall, and it takes time for the cell to make that macolic acid. So usually the Microbacterium are slow growing. Now the generation time is stated, like for E. coli, it can be as fast as 20 minutes, but the per generation is understood. So the generation time is 20 minutes per generation. And what gives students the confusion is, I guess you're used to like miles per hour. The generation time is the time per generation. Okay, it's not the generations per time. It's the time per generation. You got that? So I've stated the generation time is 20 minutes, but remember that I mean it's 20 minutes per generation. When we're talking about the generation time, microbiologists like to use semi-log plots. If they were to use a regular arithmetic plot, then on the x-axis we have time, and then on the y-axis the number of cells. And if we have the number of cells in a normal arithmetic fashion, then the growth of the bacteria will be like this in a log scale. And then one cell will split into two, two cells into four, four into eight, eight into 16, 16 into 32, so that we have a whole bunch of points here that are difficult to separate on the graph. And then our next point here will be on the ceiling. And that's why we don't like the arithmetic graph. We like the semi-log graph because if we apply it on a semi-log graph, the generation time is still arithmetic, meaning we have the number of generations here, but we have the log of the number of cells on the Y graph. One reason why we like the log scale is this point here, the next point on the graph will be about here instead of on the ceiling. Okay. And then these points down here will be more evenly spaced out. And so you can, I don't know, space them out, put them on the graph. They won't all be clumped together here. They'll be spaced out from here to here. And I'm assuming this point is similar to 32, but I don't know. Any question about any of that? All right. Oh, the other reason why microbiologists like the semi-log plot is that when we plot the equation for the growth, this is a very simple equation. Y equals mx plus b will be the equation for describing the growth of the cells. On the other hand, if we were to have this, it would be a very complicated mathematical formula. Y equals mx squared plus nx plus b to describe the growth of this. Okay, and so that's just much easier to work with. When we're talking about bacterial growth, and in general about any growth, generally the growth will look like this. And this is a bacterial growth curve. Normally there's a lag phase where there's no growth, and then there's exponential growth where the bacteria grow exponentially, and then the growth slows and enters the stationary phase where the population number of cells stay the same, and then the death or decline phase where the population of cells decreases. And they graphed this graph like deer that were introduced to an island off the coast of, I think it was Connecticut. Okay. The deer initially didn't reproduce after they were moved onto the island. And then they grew exponentially. The population entered stationary phase, and then they consumed all of their resources, and then the deer population decreased. So generally speaking, almost all populations follow this bacterial growth curve. What happens here with bacteria, if you're taking it from a solid culture and then moving the bacteria into a liquid culture to count the number of cells, there will initially be a lag phase. What's happening is the bacteria are making the enzymes they need during the lag phase for growing in the new media, for growing in the liquid culture. Once they have the enzymes they need for growing, they'll leave the lag phase and enter exponential growth. Why they grow exponentially? Well, initially, there's few organisms and lots of space. There's very few waste products around the cells and lots of nutrients. So the cells grow exponentially. But as the cells grow, there will be less and less space, more and more waste, and fewer nutrients around the cells. And so the growth will eventually start to slow. Eventually, the population will reach the stationary phase. In the stationary phase, there are as many cells dying as there are cells growing. The lucky cells have some nutrients around them and some space and not too much waste, and so they're growing. The unlucky cells have lots of waste around them, very little space, and very little nutrients, and they will start dying. So in the stationary phase, growth equals death. And that's why the population is stationary. In the next phase, the death or decline phase, there are more cells dying than there are cells growing. And so the population numbers decreases. Now, early in the death or decline phase, you do have some lucky cells growing, but there are more cells dying. And so the population decreases. Now, late in the death or decline phase, there are no cells growing. All the cells are either stationary or are dying. Any question about any of that? All right. When we're talking about microbial growth, there are a number of methods for measuring the growth of microbial populations. There are direct methods and there are indirect methods. Direct methods have some way to physically count the cells that are growing. Indirect methods have an indirect way for counting the cells. You could do a plate count, also called the viable plate count, and then you count the number of colonies. So you're doing a direct count. There's the filtration, which is similar to a plate count, and I'll talk about that in a little bit. There's the most probable number, and to tell you the truth, I've never done the most probable number way for getting the microbial growth. So of all the ways you can count the cells or get an estimate of the count of cells, I've used all of them except for the most probable number. There's a direct microscope count, and that is counting the cells under the microscope. An indirect method, turbidity, measuring the amount of light coming through the media of the tube. A metabolic activity, you measure the metabolism to determine the number of cells. Generally, there's more metabolic activity if there's more cells. And then the dry weight, where you dry out the cells and then weigh the cells. There will be more weight, dry weight, if there's more cells. The plate count is the most common method used in microbiology to get an estimate of the counts. And it's also the only way to get a viable plate count, meaning a viable count of the cells, which are viable, separating the viable cells from the dead cells. So this measures the number of viable cells by counting colonies. And to get a colony to grow, you have to have a viable cell. It does take 24 hours or more for the colonies to form, and the reports are usually reported not as the number of cells, but as the colony forming units, because we don't know if a colony was formed by one cell, by two cells, or by more cells. Any question about that? Usually to do the plate count, you make a serial dilution of your original sample. And then you inoculate agar plates from each of the dilution. And then you count the colonies on the plate where the number of colonies is between 25 and 250 colonies. And specifically, we're going to count that as 25 to 250 colony forming units in the sample. And then you estimate the number of bacteria from the plate, depending on what the dilution is. You can either pour the plate or do a spread plate method. I'll show you the spread plate method. You make a serial dilution. You have your original inoculum with bacteria, and you could plate that. But if it's too high in concentration, you won't be able to count anything. It'll just be confluent growth. So you can make a dilution, take 1 ml and put it into 9 mils. Now we have a 1 in 10 dilution. And then you mix that, and you take 1 mil out of that, put it into 9 mils of fresh media. And then we have another 1 to 10 dilution. 1 to 10 dilution times 1 to 10 dilution will be 1 to 100 dilution. And then you can dilute that further. Take 1 mil of that, put it into another tube, 1 to 1,000. Take 1 mil, put it into another tube, 1 to 10,000. Take 1 mil, put it into another tube, and it's 1 to 100,000. Remember, we're adding 1 mil to 9 mils for each of these tubes. And then you mix these up, meaning shake the tube, and then you plate from each of these dilutions. I'm going to discuss it with the spread plate, but you could pour it also. And then you put the bacteria from your, oops, going too far, from your dilution onto the plate. And then you spread that around the plate. And then you allow that to be absorbed for a few minutes, put it in the incubator upside down, and then let it incubate, usually overnight, and then bring it out and check for colonies. And if it's a good colony to count, you count the colonies. Okay. Spread plate is right here. The pour plate is where you pour it on, and then you move that across, let it absorb, and then count the colonies. So as you can see, in many of the dilutions, as well as the original, there would be too many cells to count. And for doing a count of these, you just say TM, TC, or TN, TC, too many to count or too numerous to count. And on some of the plates, or one of the plates, depending on how your dilutions are, there will only be a few colonies. What you want to do is count the colonies between 25 and 250. The reason why you don't want to count it from here is here we have only 4 colonies. So our mean is 4 plus or minus 1. That's plus or minus 25%. Here, I don't know, we have like 40 colonies. So our mean is 40 plus or minus 1, and that's plus or minus 1/40th of a percent. And so the mean is more accurate, at least especially its range or its deviation. And that's why we do the count from 25 to 250 rather than less than 25. Any question about any of that? All right, so that's the incubation, and then the viable plate count. Why this is called the viable count is, let's say that you had a dead cell that was put right here. It's not going to grow into a colony. So we only have 4 colonies growing that we can observe in this plate. And any of the dead cells do not form colonies. Why it's called a viable plate count. And this is the only method that we have for measuring viable cells. For a filtration, also a direct measurement, what you do is you have a solution of bacteria where the bacteria are too few in number to get from a viable plate count. So if you were to even plate from the original inoculation, you would not get 25 colonies. You would get less than 25. You may get less than 0, or not less than 0. You may get 0. So what we do is we put this through a filter, and the filter allows the media to pass and then retains the cells. And then you simply place the filter on an agar plate and allow the colonies to grow on the filter. Okay, and that's how the filtration method is used. It's very similar to the viable plate count. It's just using a filter to get your results. Any question there? And you use the filtration method whenever the bacteria are very low in numbers in your original sample. This could be used, for example, when you're testing drinking water. There will be very little bacteria in the drinking water, at least in most cities in the United States. There's a direct microscopic count where a volume of liquid containing the cells is measured and then counted under a gridded slide. The cells are then counted microscopically. This is not good for motile bacteria. And then you cannot distinguish between viable cells and dead cells. So usually you have a grid like this, and you put in the cells on this side of the grid, and then it's wicked under the grid. And the grid has specific lines on it. And the volume under the grid is usually known. And so what you do is you count the number of cells in this big square here. And then usually you multiply the number of cells here. What would that be? Something like 20. You take 20 cells and then you multiply by 10 to the 6 because this is taking 1 microliter or something like that. And no, that would be actually, it's 1 nanometer because it's 10 to the 6 1 millimeter. And so you'd say that our cells then would be 20 times 10 to the 6 cells per millimeter of our original solution that we sampled here. Did everyone get that? So the gridded slide takes a specific volume, and then you do your count. And under this one big square here, you multiply by 1 times 10 to the 6 cells per milliliter. You cannot tell if that cell is living or dead, and so it counts both living and dead cells. Any question about that? All right, so that's the direct methods I'm going to talk about. I'm not going to talk about the most probable number. Let's talk about the indirect methods, ways to measure the count of bacteria using indirect methods. One indirect method, which is very quick and I've used, is you just use a turbidity meter. You shine a light through the media, and so much light will be transmitted and it's recorded. And then you shine the tube containing the same media with bacteria in it. And wherever there's a cell, the light doesn't get through. And so you look at the absorbance here compared to here. And then you look at it under a chart, which has already been made for you. And you got the number of cells per mil. So we're simply putting the tube in a turbidity meter, getting the amount of light that's coming through, looking on the chart, and then we get the number of cells per mil from the chart. Now, obviously, someone has to set this up initially to make the chart. Any questions about the turbidity meter? This is probably the quickest way to get a count of the bacteria. Metabolic activity assumes that the bacterial metabolism product accumulates in direct proportion to the number of bacteria present. For example, if we're looking at the CO2 accumulating from the microbes, the more CO2 we have, the more bacteria we have. And you look at a chart, you determine how many cells you have depending on how much CO2 was produced. Any question about any of that? The dry weight is you spin down your cells, dry the cells, and then weigh the cells. The more cells you have, the more weight there will be, dry weight. You cannot tell in any of these methods what is a dead cell because it's going to add to the weight. Only the viable plate count counts viable cells. All right, any question about any of that? If not, that's it for chapter 6. And we're almost out of time, aren't we? Let me just start with one slide of chapter 8. The goals, know the terms, be able to describe DNA replication, RNA transcription, and protein translation, and understand the different types of mutation. Any question about any of that? Any question about anything? All right. I will see you at 6:30 for the lab. Can somebody please confirm that somebody's there?
Yes.
Okay, thank you. All right, I'll see you at 6:30.
Okay.
All right, bye.